So welcome everyone to the NPTEL course on Food Science and Technology. So this course is instructed by Professor H.N. Mishra sir, who is a professor in the Department of Agriculture and Food Engineering, IIT Kharagpur. I am Shikha Sharma and I am a research scholar in the Department of Chemical Engineering, IIT Guwahati. So this live interaction session is basically for week 9. So the assignment deadline for week 9 is 25th. So kindly submit the assignment on time. Uh, So the topics that we will be discussing in today's class will be food preservation principles. So it includes basically the traditional and modern methods of food preservation, then preservation of foods by addition of chemicals and removal of water, then biopreservation of foods, low temperature and non-thermal preservation, then alternative or advanced thermal techniques, then combined preservation technologies, and last would be food packaging. So we have lecture 41 to 45 regarding this module 9, which includes topics like 41 includes traditional food preservation and technologies, and 42 includes chemical and bio preservation of food. Then 43 includes non-thermal technologies for food preservation, and 44 includes alternate thermal technologies for food preservation, and 45 lecture includes about low temperature preservation of the food like freezing or refrigeration. So I hope you have already gone through all these lecture videos. So today we'll be discussing two numericals, and rest will be the theoretical based questions regarding these five lecture videos. And I hope that uh, if you have like so far like until module 8 you have no doubts, and if you have any doubts regarding any of the module, so please feel free to ask. So let's begin with today's topics. So let's discuss with the first question, which includes which of the following is a bacteriostatic method of food preservation: freezing, sterilization, irradiation, or canning. So Shiva is saying A, which is freezing is the bacteriostatic method. Okay, let's discuss. So he is correct. Freezing is basically a bacteriostatic method of food preservation. So our methods for food preservation, it can be basically divided into several further classes or subclasses based on the different classification methods, like preservation, like uh, if we are talking about the prevention or delaying of the microbial decomposition, so it can be divided into like bacteriostatics and bactericidal. So bacteriostatics includes low temperature drying and maintaining anaerobic conditions, whereas bactericidal includes high temperature processing methods and irradiation. And if we are discussing in terms of the prevention or delay of cell decomposition of the food. So it can be based on the food enzymes, like the methods which are deactivating the food enzymes like blanching and irradiation, and the chemical reactions, which includes basically antioxidants and packaging methods. Third is the prevention of damage because of insects and other problems. So basically this freezing, this bacteriostatic, basically it comes under when we are preventing or delaying the microbial decomposition. So uh, it, it includes two methods, bacteriostatics and bactericidal. So bacteriostatics includes low temperature processing methods like freezing. Whereas this high temperature method, which is sterilization, it comes under the bactericidal. Then irradiation also comes under bactericidal, and the canning or the high temperature methods, it will also come under bactericidal. So only low temperature method, which is freezing, is under bacteriostatic. So bactericidal means the methods and the mechanism used to actively kill the bacteria in the food to ensure safety and extend the shelf life. Whereas in bacteriostatics, it includes the methods which are inhibiting the growth and the production of the bacteria without necessarily killing them. So in this method only we are inhibiting the growth and reproduction of the bacteria, whereas in bactericidal we are killing the bacteria. So in bacteriostatic, this approach keeps bacterial population at manageable levels, ensuring that the food remains safe and self-stable for longer periods. So if we say the normal, if we discuss about the normal growth of the bacteria, so this is the normal growth of the bacteria. So it will multiply after some time, and further it will multiply. When we are using bactericidal actions, so what will happen? The bacteria will going to kill these, the antibiotic or the agent and bactericidal agents, they will going to directly kill the bacteria, and that's if the bacteria is killed, so we can discontinue the dose and we can see all the bacteria are killed in the plate. When we are using the bacteriostatic action, so what will happen? Uh, so when we are using the uh, antibiotic or bacteriostatic agent, so the bacteria will be going to die naturally after a lifespan of some time, after some time, because it is just inhibiting its growth and reproduction. So when if we continue the dose, then only the bacteria population will come down, and if we discontinue the dose, then again bacteria can multiply and regain the growth, because we are here just inhibiting the growth, we are not killing the bacteria. Whereas if we see in bactericidal, even if we are discontinuing the dose, because since the bacteria is already killed, so there will be no population of bacteria even after discontinuing of the dose. So here also from this picture we can see that bacteria is attacking in the bactericidal action is going on, so bacteria will become dead. Whereas when bacteriostatic action is there, so bacteria will be interacting with the bacteriostatic agent, and it will be inhibited, not dead. The microorganism will not going to die in this case. So freezing is basically a low temperature method of food preservation which has three stages. One is that we are cooling the product to its freezing point, which is pre-cooling or chilling stage, and then we are removing the latent heat of the crystallization, which is the uh, phase transition state, and then finally cooling the product to the final storage temperature, which is the tempering stage. So first the product is there, so it will first cool down to certain pre-cool, like certain temperature or certain freezing point, it will cool down, then the phase change or the state change will happen, and the latent heat of crystallization will be removed, and finally the product will be stored at that particular temperature or tempering stage is there if we are freezing any kind of product. So the phase transition part here in the freezing process, it involves basically the conversion of the water into the ice through the crystallization process and is the key step determining the efficiency of the process and the quality of the frozen product. So this phase transition is basically when water is converting into the ice. So the freezing, it basically going to reduce the water availability. So it will, because it will convert all the water into ice crystals. So what will going to happen? The water availability will be low. So it will dehydrate the bacterial cells and it will halt the enzymatic actions or essential enzymatic activity, which is important for the reproduction or the growth of the bacteria. So bacteria, because of that, comes into the dormant state. And because of that, the metabolic rate will go down or nearly zero, but their cytoplasmic membrane and the genetic material remains largely intact during the freezing. So when we are freezing the bacterial cell or the microbial cell, so what will going to happen? That extracellular ice crystals will form and the bacteria will going to be dehydrated, but the DNA or the cytoplasmic membrane, they will be intact into the bacteria. So that's why it is the freezing process is known as the bacteriostatic. So whenever bacteria will get the desired condition or desired um, environment, so it will again will going to grow. Whereas if we are doing sterilization or we are doing canning or we are doing irradiation, so bacteria will not going to grow again because bacteria is completely killed. Okay, so let's discuss second question. Which among the following methods primarily work on the principle of reducing water activity? Acidification, dehydration, then irradiation, and next last is the blanching. So which is the method which works on the principle of reducing water activity? So Anju is saying B, which is dehydration. Shiva also saying B, and Jitnyasa also saying B. So we got three answer as B. Let's see the correct answer. So they all are correct. So dehydration is the correct answer. So basically acidification is the control of pH method. Dehydration is basically the control of water activity or reducing the free water available in the food for microbial growth. Irradiation is basically about ionizing the ionizing radiation will be put to uh, inhibit the growth of the microorganisms, whereas blanching is a thermal processing. So there are different methods of reducing the water activity, which includes two type of basically the dehydration, which is two types, osmotic and conventional. So conventional dehydration includes basically sun drying or normal dehydration, whereas osmotic dehydration includes addition of sugar and salt or brine solution. So dehydration is basically involves the removal of water to reduce the water activity. So the food will become, it transforms the food into more convenient product or the self-stable food product if we dehydrate the food product. So dehydration means that we are lowering the water content up to 2 to 5% wet basis. Whereas in dehydration, the water can be up to 10 to 20 or 30% up to. Whereas in dehydration, the water content is just 2 to 5%. So this process can occur through different mechanisms like convective drying, conductive drying, or osmotic dehydration. So as we can see, initially the water from the surface will go and then the water from the inside of the material will start to go and finally the removal of moisture will be there with the help of the vapor diffusion or liquid diffusion. So if we see initially what happens, the intermolecular dehydration process is there, then combined water will also go, and then immobilized water, and then free water. So free water is the one which is at the top. So it will go first when we are dehydrating or drying any food product. So what happens, the free water which is at the surface, it will go first, and then it will vaporize first, and then the immobilized water, then the combined water, and the last will be the water which is attached to the molecules, or which is the bound water basically. And if we talk about the with respect to the cell, so what will happen if water is there? So there will be proper turgor pressure will be maintained. But if the water activity will go down, then what will happen? The cell will going to lose the water, and it will be in the shrinking stage, like this. So here in this question, dehydration is the correct answer. Okay, let's discuss this question. We have in column 1 different packaging technologies like vacuum packaging, active packaging, intelligent packaging, and edible packaging. And in column 2, we have the working principles like incorporate sensors to monitor food freshness. Then B is removes oxygen to slow oxidation. Then adds antimicrobial or antioxidant agents. And fourth is the uses of the consumables like biodegradable materials. So we need to match here. Shiva, Anju saying D, the correct answer here. Jitnyasa also saying D. Okay, let's see. So they all are correct. D is the correct answer here. So packaging basically it maintains product quality, it prevents product loss, and it aids in the transportation. It also allows safe storage, and it serves as a form of the product advertisement method also. So conventionally, for food packaging, glass, paper, metal, and plastics have been used for the food packaging. So basically, food. Yeah, so basically, food is being packaged for the protection and the preservation. So whatever the contamination is there, like the chemical or the microbial or from the insects or dusts or dirt, so it will going to, this packaging material will going to protect and preserve the food product basically. It also helps in the containment of the food product, which means that uh, there are different packaging materials, so it will going to help to put the food product into that during the transportation, or it improves the logistic efficiency, and it helps to communicate and marketing of the food product. So there is, we can see many things on the label or the packet of the food. So we have the brand logos, the ingredient list, nutritional facts, many other things, and it provides the convenience to the consumer. So it basically saves time because we can get ready to eat meals with the within these packaged food products. And some packaging materials, they are microwavable also, so it provides very convenience to the customer. So first is the vacuum packaging. So in vacuum packaging, what happens, we remove basically all the air from the packaging material. So removes air from the packaging environment to reduce oxygen and moisture. So these oxygen and moisture, they will going to contribute for the spoilage of the food product and the microbial growth. So they will be going to be removed. So mechanism is basically to remove the air so that vacuum can be created and oxidative reactions and the microbial growth can be reduced, and sealing integrity should be, so it should be properly sealed so that vacuum can be maintained and we can increase the shelf life of the food product. So basically it is used for the dried products like coffee, nuts, grains, and some perishable food products like meats, cheese, prepared meals, or ready to eat meals to prevent the spoilage and the freezer burn. Then is the smart packaging, which divided into two: active packaging and intelligent packaging. So active packaging is basically which includes some active agents like antimicrobial agents or modified atmosphere packaging or antioxidants. So and basically the some emitters or some basically oxygen capturers, like those, it will be included in the packaging material. So this kind of packaging material is the active packaging. So active packaging is for, like, we use essential oil, we use some kind of plant extracts. So all these basically are basically used uh, for the active packaging. So basically it extend the shelf life and improves the quality of the packaged product, because the active ingredient will going to provide some property which will going to interact with the microorganisms, and also it may like if they that is if it is a food and if it is the active packaging material, so whatever is the headspace is there, so the packaging material will going to interact with this headspace. So there can be some microorganisms or some oxygen radicals. So active packaging or active agents will going to interact with those and try to increase the shelf life or decrease the spoilage. Whereas in intelligent packaging, there can be like the ways for communicating. So it includes like the indicators. So if the product is getting spoiled, so some indicators like pH based or some indicators will be there which can, like, if pH of the product will going to change or if the temperature will going to change, so what will going to happen, it will change the color of the packaging material. So we can understand, okay, the packaging material, the food is not safe to consume now, it is spoiled. So it is basically the main action for intelligent packaging is to communicate the state of the food product. So it may include some sensors also, and it has some data carriers also. So it can have QR codes, barcodes, or electronic noses, e-tongue, and then it can have some freshness indicators like based on time, temperature, or gases based on the, if the gases composition may change, then the sensors will going to react in particular way and will going to communicate us about the state of the food product. So active packaging basically, it can basically release or absorb the substances into or from the packaged food or the headspace of the food packaging, intended to reduce the food safety hazard and retard the food spoilage by deterring degradative reactions such as the oxidations and the microbial growth. So if we see here in this example, this is the active packaging in which meat is there. So the active agents it will going to release, which will going to repel with the microorganisms. So active agents can be, suppose this is turmeric, so curcumin can be there. From the lemon, uh, lemon essential oil or other active compounds can be there. So it will going to interact with the microorganisms and decrease their growth, or sometimes it will going to prevent from the oxidation also. Oxidation and microorganisms growth. So it will going to try, this active packaging will going to try that the food will remain in the fresh state. So here in this diagram, this is basically the polymer matrix, which is with the either the essential oil or the plant extract. So these essential oils and the plant extracts, they are having the active in agents. So what we're going to do? So the bacteria which is trying to adhere to the polymer surface from here. So the active agents, they will try, which is incorporated into the packaging material, which is over this food product. So they will going to interact and try to kill this bacterial cell and try to stop the this adherence of the bacteria, or it will kill or the bacteria. Next is the intelligent packaging. So intelligent packaging, it enables food packaging to monitor basically the condition of the packaged food or the environment surrounding the foods through tailored sensing system. So it will going to basically tell the whether the food is in which state is there, if it is in the consumable state or non-consumable state. So it will going to monitor the condition of the packaged foods or the environment surrounding food through tailoring the sensing systems. So sensing systems will going to sense and will let us know whether the food is in the which state. So which is installed on the packaging material. So it will going to communicate the information basically regarding the food safety and the quality with the help of the array of signals. So there will be some different signals it will produce, so we can know. So in this example, like this is the food product, so whatever the headspace is there, so this indicator will going to check that and will going to tell. So suppose if the pH of the food product is changing, so there may be some like anthocyanins or some other polyphenols which will going to change the color based on the pH change, which will going to be assessed by the signals or the sensing systems to monitor the quality of the food. So for example, freshness indicators that are embedded inside the intelligent packaging can report food freshness by detecting the change in color or change in pH, change in temperature, moisture, or level of the microbial metabolites or chemicals which microorganisms produced, which are related to the food spoilage, like carbon dioxide content. It can detect, the sensors will going to detect all these things like organic acids amount, biogenic amines, volatile nitrogen compounds, or sulfuric compounds. So they will going to sense all these compounds and will communicate us whether about the food safety. Then next is the edible packaging. So edible packaging is basically, it can be edible coating and film. So it can be a basically thin layer of the any material which is we are putting on any food product, which can be basically in the form of the coating or the film like this, and it can be whether eaten together with the food or without removal, like we can eat it with the food product, or also we can remove the packaging material and then we can consume the food product, because it is edible packaging. So edible word itself saying that the packaging is made from such material which can be, we can eat, the humans can eat. So it should made from the material which are in the category of generally recognized as safe, GRAS category as per FDA. So suppose this is our food product and over this there is a layer of edible packaging material or edible coating, so or film. So what will it will going to do? It will basically going to protect this particular food from all the mechanical damages, uh, the chemical damage, physical damage, or like if there is UV light, so it will going to protect from the UV light damage, and also it will going to basically form a semi-permeable membrane. Like this structure, it will form over the food surface, which will control the diffusion or migration of the gases and also water vapor from which are going into the food and coming out towards the environment. So it will going to control the migration through this property of controlled diffusion or controlling the migration of these compounds. So that way it will going to enhance the shelf life. Also, we can incorporate some important or some fortifying agents or nutraceutical agents which will going to help to like vitamin D fortification we can do by this coating or in this coating material. So the now the food will going to be or the any suppose over any fruit we are doing this coating, so that fruit is now enriched with vitamin D, or we can this way we can fortify the food products as well with this type of packaging materials. So here vacuum packaging is related to removal of the oxygen to slow oxidation. Then active packaging is related to addition of the antimicrobial or antioxidant agents. And then intelligent packaging is basically related to incorporation of the sensors to monitor food freshness, and edible packaging uses consumables, biodegradable materials. Okay, let's see this question. Which of the following microorganisms is primarily targeted by using sorbates to preserve it? Clostridium botulinum, yeast and mold, salmonella, and lactobacillus. Anyone who would like to answer this question? Shiva B, yeast and molds. Okay, let's see the answer. So he is correct. Yeast and mold is the correct answer. So basically, here we are discussing about the chemical preservatives. So chemical preservatives are the substances which is capable of inhibiting, retarding, or arresting the microbial spoilage, microbial fermentation, acidification, and other decomposition of the foods. So its mechanism of action is that chemical preservatives, they will going to find basically interfere with the cell membrane of the microorganism or its enzymes or its genetic mechanisms. So it will going to this way preserve the food quality. Now it can be four types like antimicrobial, antioxidants, acidification, and water activity reduction. So these four mechanisms can be done by the chemical preservatives. So here this sorbate is basically the antimicrobial, it is performing antimicrobial action. So it preserves or kills uh, it basically it is going to kill or inhibit the growth of the bacteria, yeast, and molds. This way it is going to preserve the food quality. Other mechanism can be some some this uh, preservatives, they are going to perform as an activity of antioxidant activity. So they basically prevent the oxidation and that's thereby so they reduce the rancidity problem and degradation problem with the oxidation process. So some chemical preservatives, they can basically lower down the pH of the food so that the microorganism will not going to grow in the low pH condition. And some chemical preservatives, they reduce the moisture or bind with the water so that the microorganism will not going to get water for its reactions. So the chemical preservatives, they can be of class 1 and class 2. So basically antimicrobial agents, acidulants and antioxidants, they are in the class 2. So these sorbates, which is basically we use in the food potassium sorbate, it is in this class 2 preservatives, antimicrobial agents. So it inhibits the growth of the molds, yeast and some bacteria, and it is used like it is permitted by the FSSAI for use in baked goods, beverages and cheese. So this sorbate, what it basically does is, in the undissociated form or the protonated form, it diffuses across the microbial cell membrane. So it will, in the undissociated form, it will go inside the microbial cell, and it inhibits the key enzymes which are in, go inside the microbial cell because it is lipophilic in nature. So since the cell membrane of the microorganism, it is also having the lipid content. So since this sorbate is lipophilic in nature, so it is going to go inside the cell, and it will going to accumulate intercellularly. So it will going to accumulate inside the cell of the microorganism and lower the intercellular pH there. So here we can see that potassium sorbate, it is basically interacting with the microorganisms and it is inactivating the microorganisms because it can go inside the cell and lower the pH. So it will also going to inhibit the key enzymes which is important for the glycolysis process to happen and citric acid cycle. So it will going to hurt the energy production and the nutrients metabolism, and it is going to primarily affect yeast, molds and some bacteria. It also alters the cell membrane integrity, and it impairs the transport functions and the proton motive forces, and it basically prevents the spore germination and proliferation, and works in the acidic conditions. So its efficacy is more in the pH less than 6.5, where 74% remains undissociated. So this way the sorbate will going to interact with the, since this is in the undissociated form, this will going inside the microorganism, it will go inside and it will going to lower the pH of the microbial cell, and since microbial cell pH is low, so all the enzymes and the important functions, it will going to, this microbial cell will going to lose, and thereby its death will be there, the cell death will be there because of the enzyme inactivation, spores inactivation and cell wall disruption. So here yeast and mold is the correct answer. Okay, so we need to tell what are the benefits of biopreservation of food. So is it improving the safety, reduce flavor alterations, environmental friendly and shortens the shelf life? Anyone who would like to answer this question? Anju 1, 2, and 3. Shiva 3, which is 1, 2, and 3. Okay, let's see. So it is basically associated with 1 and 3. So basically, there are different methods of biopreservation like fermentation, microbial culture, bacteriocins, and enzymes. So biopreservation is basically for extending the shelf life and maintaining the quality by using the natural processes or natural organisms or their products, like we are using the beneficial microorganisms sometimes or enzymes and antimicrobial compounds so that the spoilage can be prevented and the harmful microorganism growth can be inhibited. So these are the four methods by which we do this biopreservation. So it basically works on this four principle that it produce the antimicrobial compounds. So beneficial microorganisms, they will going to produce some lactic acids, like they will produce ethanol or alcohols or bacteriocin compounds or antimicrobial compounds so that the harmful bacteria or the pathogen can be inhibited, their growth can be inhibited. So it improves the safety, which is true, and it increases the shelf life, which is here in fourth. It shorten the shelf life, which is wrong. And then because there is another mechanism which is fermentation, so fermentation will going to lower the pH of the food and creates the acidic environment which will going to inhibit the growth of the harmful bacteria. And then competitive exclusion can be there from the, so beneficial microorganisms, they will outcompete with the pathogenic microorganisms for their nutrition and space. So the pathogen growth can be inhibited by this competitive exclusion. Then enzymatic activity. So enzymes, they basically break down the microbial cell wall or other critical structure, making the microorganisms to grow. So this biopreservation methods, they basically are the natural preservative methods. So they utilizes the natural processes or the organisms which will going to avoid the synthetic chemicals and the additives. And they have the health benefits. So since we are using the fermented food products, so they can include probiotics and other health benefits which will going to improve the digestion and immune function. Then they are environmental friendly and sustainable compared to the chemical preservatives. Then they enhance the flavor because we are using the fermentation method. So it can also enhance texture, nutrition value of the food product, and then it provides the safety of the food product by preventing the growth of the harmful bacteria, and it reduces overall foodborne illnesses. So here, so it increases the safety and it increases the shelf life, and it is environmental friendly compared to the chemical preservatives, and it does the flavor and alteration. It basically changes the flavor and the texture because we use fermentation method. So it is wrong. So 1 and 3 is correct here. These are the benefits of biopreservation of food. Okay, let's discuss about the true statement for niacin as a biopreservative. So effective against gram-positive bacteria, then used in dairy products, inactivates yeast and Fourth is heat stable. So anyone who would like to tell the correct answer? Niacin as a biopreservative. Okay, let's see. So it is 1, 2, and 4 is the correct answer. So biocin is basically a bacteriocin. So it is basically a one peptide antimicrobial composed of 34 amino acids. So the primary structure is given here. So it has basically 34 amino acids, uh, and it has a positive charge of +4, and it is one peptide chain like this, and it is formed from this Lactococcus lactis subspecies lactis. It produces niacin, and also it produces lacticin also, lacticin 3147. So these two are the most extensively characterized lantibiotics. So basically, this is produced by Lactococcus lactis subspecies lactis. So niacin basically has a wide spectrum of activity against gram-positive bacteria. So it is effective against gram-positive bacteria like Staphylococcus aureus, Listeria monocytogenes, Clostridium species. So it is only bacteriocin which is permitted by the food authorities. So this peptide is basically added in the list of food additives under the European number E234. So this is only the bacteriocin which is permitted. So niacin basically binds basically the action of mechanism of the niacin action is it is going to bind with the lipid to a cell wall precursors in the gram positive bacteria. So it is going to bind with the gram positive bacteria cell wall and forming the pores there in the cytoplasmic membrane, and that will going to dissipate the proton motive force and cause the cell leakage. So this dual action of pore formation and inhibition of the peptidoglycan synthesis lead to the rapid bactericidal effect against the pathogens. So this is the niacin molecule. So it is going to basically bind with the lipid 2 from in the bacterial cell wall, and it has basically divided into two parts. One is recognition module and second is membrane active module based on the pattern of interaction with the bacteria. So this is the recognition module and this is membrane active module, and in between is the this one is the hinge region. So this is divided into this two module. So either this will going to act with this recognition module or adhere with this recognition module to the lipid 2 protein of the cell membrane, or it will going to react with the membrane active module with the microbial cell. And this way it will going to enter into the microorganism or microbial cell. It will going to form the pores there, and also it will going to inhibit the so pores, if the pore is there in the microbial cell, so the components which are inside the microbial cell, it will going to come out and leakage will be there. So one action of uh, like inhibiting the microorganism is pore formation and leak, and forming like creating the leakage from the microbial cell. and another is the inhibition of the peptidoglycan synthesis, which will both going to act on the bacteria and kill the bacteria. So here, since it is effective against the gram-positive bacteria and it is used in the dairy products, and it basically inhibits the gram-positive bacteria. So it doesn't inactivates the yeast and is also heat stable. So 1, 2, and 4 is the correct for niacin. Let's see this question. So which of the following statement is correct with respect to the using antioxidants in the food? So statement 1 is that antioxidant will going to prevent the lipid oxidation. Then antioxidant will inactivate the enzymes responsible for spoilage. Then BHT and BHA are the common synthetically used antioxidants, and antioxidants are effective only in oxygen free environment. Anyone who would like to answer this question? Okay, let's see the answer. So here 1 and 3 are correct. So antioxidants are basically they are inhibiting the lipid oxidation because they will going to scavenge the free radical intermediates, and they will going to also sometimes chelate the pro oxidant metal ions. So overall, they will slow down the oxidation process or auto-oxidation of the chain reaction that will going to lead for the rancidity in the fats and the oils. So in the last class also we discussed that antioxidants, they will going to scavenge these free radicals from the lipids or from the fats, and they will going to reduce the rancidity problem. So statement 1 is correct. Then BHA and BHT. So BHA is E320 permitted additive. So BHT is E321, 320 and 321. So they are basically the permitted additive to be used in the food. So they are basically well established synthetic polyphenolic antioxidants which are used in the range of the food products like oils, snacks, meats, baked goods, and they are basically used to inhibit the lipid oxidation, and they function mainly by the chain breaking antioxidants by donating H plus atom. So they will going to donate H plus atom to the lipid species. So lipid species will be there. So lipid peroxide radicals will be there, and to this H plus ion they will going to donate, and thereby terminating the propagation of the oxidation reaction. So thereby it will terminate the oxidation reaction and prevents the rancidity of the fats or the oil or any meat or baked goods like that. So statement 3 also correct. So antioxidants are basically designed to work in the environment which is having oxygen. That's why if there is no oxygen, so there will be no oxidation process will be there. So why we will use antioxidants? So basically these antioxidants, they are basically designed specifically so that they can prevent the food spoilage in the oxygen rich environment. Some antioxidant compounds like, so this statement is wrong because they work in oxygen presence, and this statement 2 is actually partially correct and partially incorrect. Why I am saying so? Because maximum, like if we are taking this BHA, BHT, so or sorbates, these will going to these antioxidant compounds or the chemical preservatives. They will work they'll work on not on the principle of antioxidants, but some antioxidants, sorry, they will work as an antioxidant, but they're not going to inhibit the enzymes. So here it is saying inactive enzymes, but maximum antioxidant, they will not going to inhibit the enzymes, they will going to inhibit the growth of the microorganisms. So whereas this sulfur containing antioxidants, which are sulfites and the ascorbic acid, they will actually inhibit or they will going to interfere with the spoilage related enzymes like polyphenol oxidase and the peroxidase by altering the enzymes redox environment, and sometimes competing for the substrate and competing or binding to the active sites residues. So these ascorbic acids and the sulfur containing antioxidant, they will inhibit or they will interact, they will compete with the enzymes of the microorganisms. And so this statement is correct for only sulfur containing antioxidants and ascorbic acid, but the other antioxidants like the benzoate or sulfonates or sorry, sorbates, benzoates and BHA, BHT, they will actually not going to interact with any enzyme, and they will not going to inhibit the enzyme. They will directly inhibit with the microbial cell. So it is partially correct and like partially wrong, partially right. Whereas a baki or the statement 1 is totally correct. Statement 3 is also correct. Statement 4 is incorrect completely. Okay, now let's discuss this question about pulse electric field. So statement 1 is saying that the pulse electric field treatment disrupts the microbial membrane through electroporation. And statement 2 is saying that the pulse electric field treatment is primarily suitable for solid food products. So we need to tell statement is correct. Anyone who would like to answer this question about pulse electric field? Okay, so only one statement is correct here. So pulse electric field is basically a technique in which the food is going to be placed between the two electrodes like this. So the food will be between two electrodes. And exposed to the short bursts, which is less than 1 second of high voltage electric field like 10 to 80 kilowatt per centimeter. So very high voltage of electric field will be applied and uh, for a very short case. So this is basically a non-thermal method, is conducted at some ambient and slightly above ambient temperature, and it effectively disrupts the microbial cell membrane, leading to microbial inactivation while preserving the food taste, flavor, color and freshness. And it is highly effective on the cellular proteins and inactivates the microbial cell and enzymes. So here, before treatment, the cell is like that. After the applying of the pulse electric field for uh, short time and high voltage, so the cell will be like this. So there will be poration will be, we can see here poration is there due to application of high electric field, which will going to result to the loss of the cell integrity and loss of the cell fluids. So one mechanism of action is electroporation is happening here. So when the cell, we can see here, so when there is this alternating current or high voltage current is exposed, so cell will going to be like this, poration will be there. So exposing a biological cell like the plant, animal and the microbial cell to a high intensity electrical field, kilowatt per centimeter. So in the form of the very short pulses like microsecond to millisecond, induces the formation of temporary or permanent poration on the cell membrane. This will going to lead to the leakage of the cellular content and eventually the cell will die. So it will basically lead to the permeation of the cell membrane. So it will going to increase basically the permeability of the cell membrane, and if the intensity is very high, so the cell membrane will going to disintegrate. So basically what is happening, the mechanism behind electroporation is that there is an increase in transmembrane potential, electromechanical compression of the cell membrane and conformational changes in the lipids or the protein molecules. So basically, theory behind this is dielectric breakdown theory. What it says is that the main cause of the cell membrane destabilization is because of the increase of the transmembrane potential. So why the poration is happening or the permeation is happening? Because of increase in the transmembrane potential. What it means is that the potential or the high transmembrane potential is developed. So what is happening basically, the cell membrane, it maintains an electrical and chemical gradient, which is known as electrochemical gradient, between the outside and the inside of the cell. So we can see here that the cell is basically maintaining some kind of electrochemical gradient here. So the electrical gradient is due to the excess of the negative charge. So negative, so like this, if there is positive charge, negative charge will be there. So what will going to happen from the cell here, negative charge will going to accumulate inside the microbial cell. Here positive charge will going to accumulate. Here positive outside, here outside the microbial cell, negative here ions will be there. So because of this positive and negative, negative and positive, there will be this transmembrane potential is developed. So when this electrochemical or gradient will be very high, so the very high negative ions will accumulate immediately at the inner surface of the cell membrane. So inside the cell membrane, if the higher chemical electrochemical gradient will be there, so high negative charge will accumulate here, and it will going to be outside, equal positive charge will accumulate here immediately outside the cell. So this will going to establish across the cell membrane, and there will be some transmembrane potential will develop. So if uh, so it is around 0.1 volt. So when we it is around 0.1 volt, this transmembrane potential we call is a resting potential. But when we are applying very high electric field, so the ions in the cell moves along the electric field to the membrane surface. So because of the this free charges will accumulate on both sides of the membrane, and the accumulation of these surface charges results in an increase in potential across the membrane, so which will going to create the electric field which is induces an additional transmembrane potential which will be larger than cell's natural potential. So if we are applying external electric field, so what will going to happen? Because of external electric field, the more number of the ions will accumulate inside the inside this cell of the microorganisms, and outside more positive charge will be there. So because of that, there is high amount of the potential will develop, which is more than the resting potential or the threshold potential, which is more than the resting potential. And after it will develop more, and after some time it will come to the threshold potential. And after that, if more potential will be there, then there will be poration in, because of the more development of the transmembrane potential. So the induced transmembrane potential, this, which is the difference between the intra and extracellular potentials, which is internal of the cell like this and external like this. So is unevenly distributed over the surface of the cell membrane, and that is the potential difference is high on the membrane sites close to the electrodes, and the low as the distance between the membrane sites and the electrodes increases. So there is the breakdown of the cell membrane when there will be this overall transmembrane potential, which is the sum of the resting potential and the induced potential, when it will reach a threshold value, which is a critical membrane potential. When the overall transmembrane potential increases the threshold value, it will going to break the cell membrane, because there will be more positive negative, so there will be going to attract more. So if there is more attraction, it will going to lead to the compression pressure on the cell wall of the microbial cell. So that will cause the decrease of the membrane thickness. So if there is this positive and negative charge more build up, here negative, here positive, they will going to attract each other, and because they are attracting each other, there will be decrease in the membrane thickness due to the compression pressure will be there. So this compression of the membrane, it creates elastic strain forces. When the electro compressive forces will exceed the membrane elastic strength, membrane breakdown will be there, or the progression or the pore formation will be there. So if the size and the number of the pores becomes large, and in relation to the membrane surface, there will be this irreversible breakdown beyond certain level, and there will be the associated mechanical destruction of the cell occurs. So this basically, because here more positive charge, more negative charge, so they will going to attract, and because they are attracting each other, this cell wall or the cell membrane of the microorganism, it will going to be compressed or become thin and thin and thin. After beyond certain point, it will break down this like this. There will be pore formation. More pore is there, so between is more, so that will become irreversible. So here statement 1 is saying that the pulse electric field treatment disrupts microbial membrane through electroporation, which is correct, and it is basically suitable for the no, primarily for the solid food products, which is wrong, because I think it is mostly used for the liquid food products. Okay, let's see the next question, that the statement which is not correct with regarding to the plasma treatment of the food preservation, that the plasma generates reactive ions and free radicals to inactivate the microorganisms. Plasma is effective for surface sterilization of fresh produce, and plasma treatment is highly efficient or effective in inactivating the bacterial spores, and the plasma operates at low temperatures, preserving the food quality. Anyone who would like to answer this question? Jitnyasa D. Okay, let's see. So here C is correct because that plasma is basically the fourth state of the matter where the materials has become so hot that these electrons are no longer bound to the individual nuclei. So it will electrons will come out the nucleus, and this plasma is basically an ionized gas which will have free ions, free electrons, ions, reactive atoms, neutral molecules, and photons. So plasma is having all these uh, things, like it can have positive ions, negative ions, electrons, metastables, atoms, free radicals, photons. On average, this plasma is the fourth state of matter. So it is basically the electrically neutral because any charge unbalance would result in electric field that would tend to move the charges in such a way that it will neutralize the overall charges of the opposite sign. So it is known as a quasi-neutral gas. It is generated by an electrical discharge of gases such as argon, helium, nitrogen, oxygen, and atmospheric air. And it can be hot plasma, cold plasma, and it can be under atmospheric condition or high or low uh, pressure conditions also. So plasma basically generates ions and free radicals to inactivate microorganisms, which is correct, and it operates at low temperature, preserving the food quality, which is also correct because it can have we can have cold plasma treatment also. So the overall mechanism of action is basically, so it inactivates the microorganisms. So cold plasma produces reactive species that damage the microbial cell wall, proteins, and DNA, and effectively inactivate or kill the bacteria, yeast, and mold and other microorganisms on the food surfaces. So what it this is for second mechanism is surface modification. So reactive species in the cold plasma, it can enhance the surface properties of the packaging material, which improves the wettability, adhesion, and barrier properties to better preserve the packaged food. And third is the chemical degradation. So cold plasma can break down undesirable compounds basically in the food like pesticides, contaminates of flavor compounds, which improves the overall quality and the safety of the food product. So here, this is an example where atmospheric cold plasma is being applied, where the gas molecules basically, here, these are the oxygen, nitrogen, and CO2. These gas molecules are ionized in an electric field. So this is an electric field where this is ionization is happening. So here, the mixture of electrons and the ions are produced, and then these charged carriers will going to excite and create a large quantity new charged materials they will create. Like this plasma is a like mixture of all these compounds or all these ions and the electrons like this. All these ionization will going to happen here. So we can see here, this is a first one is a dielectric barrier discharge. So here between two electrodes, the plasma is being created. And this is atmospheric plasma jet. So here, with the help of this nozzle, the plasma is exposed to the open environment. So here, if we see that plasma is effective for surface sterilization of fresh produce, which is also correct. And plasma treatment is highly effective in inactivating the bacterial spores, which is wrong because it basically inactivates the microorganisms cells, uh, but not very highly effective for bacterial spores. So statement 3, option 3 is incorrect here. So next question is the dose of ionizing radiation for food preservation is expressed in. So what is the unit in which we express the doses of ionizing radiation? So Anju is saying D, gray. So she is correct. Basically, the international unit of absorbed dose is basically gray, GY, which is equivalent to 1 joule of the energy per kg of the material. And we can see this kind of the sign on the food packet where we are using the food. We are using this radiation or ionizing radiation irradiation method for preserving the food. Okay, let's discuss this question. In column 1, we have the non-thermal preservation technologies like HPP, irradiation, PEF, and plasma. And in column 2, we are having some working principles like ionization of DNA and cellular structures, electroporation of microbial cell membrane, C, inactivation of pathogens using reactive ions, D, uniform pressure application in all direction. So we already discussed about pulse electric field and plasma treatment in detail. And so I think this question is easy to answer now. Anyone who would like to answer this question? Anju, B. Okay, so she's correct. So here, uh, high pressure treatment is associated with the uniform pressure application in all direction. Then irradiation is ionization of DNA and cellular structures. Pulse electric field is related to electroporation of the cell membranes, which we discussed already, and the plasma is related to inactivation of the pathogens using reactive ions, which is also like reactive ions, or we use electrons and the protons and many ions, uh, which will going to, or the plasma, which will going to inactivate the pathogens. So HPP is basically where we are using high pressure like 100 to 1000 megapascal and we will apply for this pressure for 30 seconds to 30 minutes. We'll hold this pressure for like this. So we will have some food product, and this pressure will going to be applied through this medium on this food product. So the food product can be used basically with or without packaging, and if there is some packaging, so it should be a flexible kind of packaging where the volume like 10 to 15% change in the volume it can sustain basically when we are applying the high pressure. So pressure medium can be water, castor oil, silicon oil, glycol, or ethanol, and it works on the two principle isostatic principle and Leach-Eteriol's principle. So isostatic principle means that whatever pressure we are applying is instantaneous and it is uniform throughout the sample. So the pressure we are applying is all uniform and instantaneous in the sample. And Leach-Eteriol's principle is saying that reactions resulting in a volume change are influenced by pressure applications. So because we are applying the pressure, So volume change can be there. So suppose like we are plotting this, so we can see that here, if we see here in this diagram, so what we are doing is we are putting some kind of the pressure with the piston, and this is the where the pressure is applying. So here our sample will be there with the pressure medium, and the food is getting the HPP treatment. So initially what happens, the pressure will increase and we'll hold the pressure for some time and the pressure will decrease. Like this it happens. So because the pressure is increasing, so what is happening, some temperature will also increase and will be there and will decrease. So this is the, so what is there, what first we have the, this is the come up time, then this is a holding, and this is the decompression cycle. So here high pressure is being applied, so that is causing the death of the microorganism due to permeabilization of the cell membrane. Then there is the changes reversible happening at the low pressure and irreversible at the high pressure. Then there is disruption of the protein structure and the cell membrane and reduction of the enzyme activity, which ensures high quality and self-stable product, and only non-covalent bonds will be affected. So the organoleptic properties will going to be unaltered in HPP. So this HPP is combined with sometime other methods like antimicrobials, heat, so that the effectiveness can be improved and the processing time can be reduced. And HPP is effective against most of the microorganisms, but not all. So here in this, the pressure required to achieve a 5 log cycle inactivation is given, which is applied for 15 minutes treatment. So this much pressure, this high pressure in megapascal, we are applying this pressure. So this high pressure like 200 to 700 megapascal we are applying for 15 minutes. Then these microorganisms are getting inactivated. So sometimes what happens, we are applying the pressure, the enzymes may denature, but again it may renature. But if we are applying the pressure beyond certain, like very high pressure, so after that it will, this enzymes will denature and it will not regain their structure again. In the irradiation, what happens basically, second method is irradiation. So irradiation happens when we are exposing our food with the ionizing electromagnetic radiations like gamma or the X-rays. So electromagnetic radiation, if we see, this is a spectrum. So it includes, like it has no mass or charge, and it will have some photons that will have some energy. So the basically we see here gamma and X-rays, which are the high frequency waves we are using for irradiation process. So irradiation process happen after a food is produced or packaged. So this kind of sign we use if the food is irradiated on the packaging or the labeling. Food packet, we use this sign. So the irradiation or the radiation, it is going to penetrate the food and kill the germs or prevent them from multiplying, and it does not linger in the food. So what happens, how irradiation is formed? So this is the nuclei of the atom. So basically from this, high energy electrons produces and beta radiation produces, and high electromagnetic photons produces. So gamma radiation also produces, and from this alpha radiations also going to produce from this radioactive decay of the nucleus of atom. So basically these radiations, how and what it is going to do is they have the direct effect and indirect effect on the microorganisms. So basically these radiation, they'll have enough energy to remove tightly bound electron from the atoms. So they'll go directly, they will remove this electron and creating the ions. Ion will form, and the type of ionizing radiation uses in food irradiation, basically gamma radiation, X-ray and electron beams are basically used in the food. So direct action is that the irradiation will going to directly attack this atom, and it will free these electrons, and these electrons will going to affect the DNA of the microorganism. And another is that it basically indirect action, that this generated electron will going to interact with the other molecules like H2O, and it will form some radicals, and that radical will going to attack the DNA. So this is the two way how the microorganisms DNA is affected by the radiation. So the food will going to absorb this energy from the radiation, and it will interact with the molecules in the food and affecting the DNA and cellular structure of the microorganisms. And the sensitivity to irradiation for the food is that the water is very sensitive, then protein and other nitrogenous compounds, and then the lipids and the fats. And it we can have low dose less than 2 kilogray. So it is used for sprouting inhibition, delaying the ripening and disinfection of the stored cereals and pulses. And medium doses we apply like 1 to 10 kilogray for the reduction of spoilage microorganisms in the fish and poultry, decontamination of the herbs and spices, and reduction of the level of pathogen microorganisms, and also like similar to pasteurization process. And the high doses which is greater than 10 kilogray is used for sterilization process. So here we can see here is the pathogen. So it is going in irradiation chamber, which is this is killed, and finally we get the food product which is having less or no microorganisms. So here in this B is correct here. Okay, so let's discuss about mechanism of pulse light processing of the food, which is correct statement. So pulse light emits high intensity pulses of light that coagulate microbial proteins. And second is PLP effectively penetrates dense opaque foods for microbial inactivation. Anyone who would like to answer this question? Okay, let's see. So here neither 1 and 2 are correct. So PLP is basically a known thermal process. So it uses basically high intensity pulse of broad spectrum light to inactivate the microorganisms, and it involves exposing the food to intense bursts of light like UV light, visible near infrared, and it is going to disrupt the microbial cell structure and affect the DNA. Then basically it is an eco-friendly technology with a lower carbon footprint, but it is suitable to liquid food, restricts its versatility application in the food industry. And the mechanism is that it works with light emission and enzyme and microbial inactivation. So it basically emits high intensity light pulses about 0.5 to 20 milliseconds that will going to denature the protein of the enzymes and cell wall, membrane and DNA. So like this is the basically here is the light is there, pulse light will be there. Here food is placed, and uh, it is being exposed to the pulse light. This is a continuous flow pulse light system. Second one is, so what is happening here when the food is when the microorganism is getting exposed to this light, pulse light. So what will going to happen? The photochemical effect is there, photophysical effect is there, and photothermal effect is there. So what is happening basically? So DNA is getting affected, and also the thermal effect is there. So because of high temperature, also microorganism is getting affected, and physical photophysical effect is also there. Because of that, microorganism is getting affected. And also, like, apart from this light emission and then inactivation of microorganism and the enzyme, there is also penetration and coverage. So this wavelength of the light is basically able to penetrate the food or surface based on the which wavelength we are using. So it will also go inside the food and can deactivate the microorganism. So we were discussing this question that PLP effectively penetrates dense opaque food for microbial inactivation, which is not true. That it needs food in the direct contact so that it can and less dense food so that it can penetrate inside the food and it can inactivate the microorganism based on these three mechanisms: photochemical, photothermal, and photophysical. And pulse light emits high intensity pulses of light, which is correct, that coagulates microbial proteins. So it basically doesn't coagulate exactly, but it affects basically the enzymes, and it affects the cell wall and DNA and membrane proteins. It basically denature it, changes its structure. So, and changes the structure such that it will become inactive for the purpose what they want, like their primary purpose is there, so they're not going to able to perform. So here answer D is correct. Okay, let's discuss about mechanism of ohmic heating. So this is ohmic heating system where we are putting electrodes and power supply we are giving to the food, and it is getting heated using ohmic heating principle. So one is that statement one is saying that high thermal damage to sensitive foods, uneven heating in foods with variable densities, inability to retain food's nutritional quality, and D is lack of energy efficiency in food processing. Anyone who would like to answer, or anyone who is having any doubt so far? Okay, let's see the answer. So basically here B is the correct answer. It basically best describing the ohmic heating. So ohmic heating, which is also known as electrical resistance heating, it is a thermal processing of the food material where what is happening, which is served as an electrical resistor. So food is basically here food is we are keeping, so it is acting as a electrical resistor, and when heat is passing or electricity is passing through it, it will going to generate some heat. So an electrical energy is dissipated in the form of heat, which is going to rapidly and uniformly heat the food product. Basic principle is that we are passing the alternating electric current from these two electrodes. So these two electrodes, this one and this one, between these we are passing electrical current. alternating electrical current. So one time positive, one time negative. So here in the resistor, food is which is acting as a resistant will develop heat. So this electrical energy is conducted through the food material, which is converting that into thermal energy due to the electrical resistance of the food, which is also known as Joule's effect, and it will going to lead to the volumetric and instantaneous heating. So the mechanism is that the temperature is directly proportional to the resistance of the food and also to the current which we are providing. Electrode placement on either side of the food to apply the electrical current. So we are placing two electrodes and in between the food is there which is going to heat the food product. So this electrical current, when it is passing through the food, it will generate uniform internal heat due to its distributed resistance. So this ohmic heating, it is generated generating heat evenly inside the food product, ensuring that the uniform temperature is there in the food. So consistent processing by temperature control we can do by adjusting the electrical current and time. Here, to calculate the power, we can use power equals to current, P equals to I square into R. So, since also it can also be written as electrical conductivity into electrical field strength square. So here the heat is generated which is depending on the electrical conductivity or resistance of the food material, but the food is not uniform. Food can have moisture, salt, its pH can be different and the density will be different. So based on these regions of different composition, the density will be different and the power density will going to be different. So this will going to lead to the non-uniform heating of the food because food is a heterogeneous. So example, if the particulate liquid mixture is there, food with varying tissue density is there. So the local density, if it's different, the power density will going to be different because it is depending on the electrical conductivity, which is depending on the different heterogeneous zone of the food. So heterogeneity in the density and composition causes the localized hotter and cooler zones, which is why heating pattern is often described as uneven in such complex food products or ohmic heating. So the best describing is B, which is that uneven heating in the food with variable density. Next is this, which is on radio frequency heating. So we need to tell the correct statement with respect to the energy efficiency. So statement 1 is saying that RF heating minimizes energy loss during the processing. And second is RF heating uses higher energy than infrared methods. Anyone who would like to answer this question? Okay, so answer only statement 1 is correct here. So dielectric heating basically can be basically two. So dielectric heating or basically the method which are based on this dielectric heating or dielectric energy consumption are two. One is radio frequency, another is microwave, which we'll discuss after this. So dielectric drying is basically uses dielectric energy for removal of water. So what is dielectric drying? It uses basically electromagnetic waves to generate heat within the food material due to two methods, which is dipolar relaxation and ionic conduction. So what is dipolar relaxation? It means that water molecules which are there in the food, it is going to like act as a dipole when electric field is applied, and they will rotate at very high frequency. So when the electric field is applied to the food, the water molecule, it will going to act as a dipole, and it will uh, if is positive charge is there, it will form positive, here is negative, and it will repel, and it will go towards negative, and this negative will go towards positive. And since we are using alternating current, so it will continuously change. Second is the ionic conduction, which is ions which is there in the food, like salty foods, will migrate under the influence of electric field and generating the heat. So that means the ions here in this case, not this water molecule, but the ions will going to act like this and generate the electricity. So the radio frequency, it will usually work under 1 megahertz to 100 megahertz, and it targets the polar molecules and ions to generate heat, and it involves the transfer of electromagnetic energy directly into the food, and it initiates the volumetric heating due to frictional interaction between the molecules. So if we see infrared, it is here, and if we see radio frequency, it is here. So it is at higher frequency. So RF heating uses higher energy than infrared methods, which is it doesn't use higher energy, but it minimizes the energy loss during the process, which is correct because it is energy efficient method. Also, this RF heating is also known as high frequency dielectric heating. So here, what is happening? Like this, if we see, this is dipolar rotation. So water molecules will have formed dipoles, and it will align like this in the when electric field is applied. And ions which are there in the salty food, it will be distributed randomly, and when the electric field is applied, it will be aligned. It will come and align. So there will be, because the oscillating electric field will be there between these two electrodes, so all the dipoles, which is this, and all the ions, they will continuously oscillate based on the or realign based on the electric field. That's why they are going to generate some frictional forces, and thereby heating of the food is done. So during RF heating, the product is going to be heated, forms a dielectric between two metal capacitor plates which are alternatively charged positively and negatively by a high frequency alternating electric field. The polar molecule like the water try to align themselves with the polarity of electric field. Since the polarity changes like at 27 megahertz, like 27 million times per second, the polarity is changing. Suppose, so the molecule will try to continuously realign themselves with this high electric field by flipping and flopping motion. So the resulting is the high kinetic energy and the friction, which will, because of the colliding neighbors molecules, generate the heat in the food product. So the RF operating principle is same as microwave heating, but the operating frequency in RF is lower than which is 27 megahertz. In microwave, it is 2450 megahertz. So in RF, the thickness of food is restricted because of the distance between the capacitor plates. So we can use a particular thickness material only, and RF energy can be used to heat or evaporate moisture from the product. Okay, let's discuss this question. In column 1, we have different preservation technologies like ohmic heating, microwave heating, infrared heating, and refractance window drying. And in column 2, we have the working principles. So A is heat transfer via electromagnetic waves, And then second, B is direct heating through electrical resistance. C is that surface heating using infrared radiation, and D is drying using infrared radiation through a transparent substrate. Anyone who would like to answer this question? Anyway, we already discussed about uh, ohmic heating. Also, we discussed some part of microwave, which is similar basically to the radio frequency, but the frequency or the operating frequency is different. We also discussed some part of, no, we discussed this only, we discussed ohmic heating. So Anju is saying A here in this case. Let's see. She's correct basically. So microwave heating uses, as we discussed, that it also uses the dielectric component of the food which interacts with the electromagnetic waves. So if we are using the domestic use, it will be 2.45 gigahertz, and for commercial use, 915 megahertz. So here we will have our food, and from top, the microwaves will come and interact with the food. So heat generation will be based on two principles: dipole motion, rotation, and ionic conduction. As we discussed in case of the radio frequency, the same will be there, and it will also generate the volumetric heating of the material. And because of the rapidly moving of the dipoles and the ions, so the high amount of the friction force will be generated. Infrared heating is basically infrared is the portion of the in the electromagnetic spectrum which is here, if we see, which falls between the region of visible and microwave. So when electromagnetic radiation will come and interact with the food, it causes changes in the electronic, rotational, and vibrational state of the atoms and the molecules. So after penetration in the food material, IR radiation, it will vibrate the constitutional molecules at around very high frequency, around 60,000 to 1,50,000 megahertz, and it will result in the intermolecular friction and bring about rapid and internal heating. So IR infrared will come and interact with the food, and since it is at very high frequency, so it will going to reduce the intermolecular friction and rapid heating. So this is because this OH bond in the water, it is going to absorb this IR radiation and like this, and it is going to rotate and vibrate at this high frequency. So it is going to reduce the friction. Refractive window drying, it is basically a thermal technology which is using the infrared radiation to remove the moisture content from the food. So what happened, we put our food and we'll spread into the thin layer on a heat resistance and transparent film. So infrared will going to come and remove the moisture will go out like this. So this is basically the mechanism. So RF drying process, basically it uses the infrared mechanism. So it also, it depends on the product thickness, the temperature of the product initial, and how much time we are taking, and how much the film uh, thickness is there. So based on that, it will work. So one, which is ohmic heating, is related to direct heating through electrical resistance. Microwave heating is related to heat transfer via electromagnetic waves, and infrared heating is basically surface heating using infrared radiation, and refractive window drying is the drying using infrared radiation through a transparent substrate. So what are the advantage of this refractor window drying method? Anyone who would like to tell? So the advantages are that it is energy efficient and it retains nutrition in the dried products. So it basically uh, have the it will retain the quality, the color, flavor, and the nutritional value of the food product. And it is an energy efficient process because it produces like more. Basically, it is more energy efficient method because it is a direct heating the moisture in the food, reducing the energy loss, and it reduces the process time. So the thin layer of the food is there, so it will be going to fast dry the food compared to the traditional method. And it is limited to drying only fruits. So it is wrong because we can dry many things like probiotics, we can use for encapsulation, fruits and vegetables drying, herbs and spices drying, and we can use puree and prepared foods for drying and nutritional supplements also. So it is wrong. And preservation of the volatile compounds. So basically, if we compare with the conventional method, it uh, do preserve volatile compounds and it is used for herbs and spices for and it will help to retain their volatile compounds and essential oils. But if we compare it with the freeze drying method, it is not that effective to preserve the volatile compounds. So only 1 and 2 are correct here. Statement 1 and 2, which is energy efficient and nutritional retention in dried products, are correct. Okay, let's solve this question. A storage facility has 20 containers and each weighing 5 kg with specific heat. CP is 2.1 kilojoule per kg per Kelvin. The temperature difference between the ambient and storage is 25 K Kelvin and the cooling process is completed in 5 hours. Calculate the heat removed from the container in joule per second. Anyone who would like to calculate this? Okay. So since we have this mass, the total number of containers, it is given 20, and each container is of 5 kg. So we can calculate the total mass of all the containers. So total mass of all the containers M is equals to 20 into 5, which is 100 kg. And the specific heat is given as 2.1 kilojoule per kg per Kelvin. So which I can write as or we can write as 2100 joule per Kelvin. So I am converting kilojoule into joule here by multiplying 1000. So 2.1 into 1000 would be 2100 joule per kg Kelvin. And the temperature difference del T between the ambient and storage is given as 25 K. And time took for cooling is given as 5 hours here. So 5 hours into second, if we want to convert 5 into 60 into 60, which is 1800 seconds. Now I can simply apply the heat removal formula for the from the containers will be Q equals to mCp delta T. So M is 100 kg, Cp is 2100 joule per kg Kelvin, and del T is given as 25 Kelvin. So it will come as 525,000 joule. So rate of heat removal from the containers will be divided by this time. So if I divided this by time, which is 18,000 seconds. So it will be 291.67 joule per second. So the heat removal from the container would be 292 joule per second. Is this question clear to everyone? Can we solve this next question? A refrigerated room has a wall with an area of 200 meter square and insulation thickness of 0.5 meter. The thermal conductivity of the insulation is 0.025 watt per meter Kelvin. The ambient temperature is 30 degrees Celsius and the chiller temperature is 5 degrees Celsius. Calculate the heat loss through the walls in joule per second. Anyone who would like to calculate? Okay, let's see. So the area is given as 200 meter square of this refrigerated room wall. And the insulation thickness is given as 0.05 meter. The thermal conductivity of this insulation is given as 0.025 watt per meter Kelvin. The ambient temperature, outside temperature is given as 30 degrees Celsius. And the chiller temperature or this refrigerated room temperature is 5 degrees Celsius. So we need to calculate the heat loss through the walls in joule per second. So we can use here Fourier's law. So Fourier's law of heat conduction says that rate of heat transfer is directly proportional to the thermal conductivity of the ice or thermal conductivity, the cross-sectional area through which the heat is being transferred, and the temperature between the heating surface and the frozen layers, but it is inversely proportional to the thickness of the frozen layer. So it is basically, this Fourier's law is basically saying that Q, which is the heat transfer, is directly proportional to the thermal conductivity, area, and the temperature difference, but it is inversely proportional to the thickness of the layer. So Q equals to Ka dt by dx. So Q equals to K, which is the thermal conductivity. So K is given as, thermal conductivity is given as 0.025. So I can write 0.025 watt per meter Kelvin. Area is given as 200 meter square. The refrigerated room temperature is 5 degrees Celsius minus ambient temperature 30, which is I am writing here in Kelvin. So 5 degrees Celsius plus 2, 7. Sorry. If we calculate the, what is the conversion? 274 or 275, I think we use for Kelvin. 275 we take because 274 point something is there. So sometime we take, so I think. What is the conversion? Anyone who would like to tell me the conversion? So if I am also taking in the degrees, into the degree Celsius, so it will be 5 refrigerated temperature minus 30. So overall it will be 25. So divide by L, which is thickness. Thickness is of the insulation is here 0.05. So 0.05. So here, if I calculate this, it will be 5 minus 30, which is minus 25. And if I will multiply 0.025 into 200, divide by 0.05, it will be 100. So 100 into minus 25, it will divide by 0.05, it will be minus 2500. So that is 2500 joule per second. Minus is because heat is flowing from low temperature refrigerated walls towards high temperature refrigerated wall. So it will be in minus. Is this question clear to everyone? The answer is A, which is 2500. Is this question clear? So 1 degree Celsius is 270. So in if we want to convert degree Celsius into Kelvin, so it will be 273.15 overall. But I'm taking here roughly 275. So I have added 275 plus 5, which is 280 here, and here 305. So I am taking the round off number, which is 275. But here actually if we are converting, it will be 273.15, 1 degree Celsius into Kelvin. But the difference is 5, which is 5 minus 30 is 25. Is this question clear to everyone? Can we move to next question? Which of the following statement is correct with respect to freezing of food? One is slow freezing leads to large ice crystal, and second is large crystals cause greater tissue damage. Anyone who would like to answer this question, or any question which is not clear so far? Anyone who would like to answer this question? So here both the statements are correct. So freezing is basically conversion of the liquid water which is into the ice, and basically since water is converted into ice, so it will be low available for microbial growth, and low water activity is there, so the microbial growth is arrested due to the cold shock. So this is the ideal for freezing curve, that we decrease the temperature. It'll the thermal arrest will be there, and finally the equilibrium freezing temperature will get. But usually food is having many different contents, like salt is there, sugar is there, many other different food components are there. So when we decrease the temperature, it will super cool, and then it will the latent heat will be removed, and finally if we continue to heat, the crystallization or the ice formation will be there, and finally the temperature or the tempering or the freezing temperature is attained. So in freezing of the tissue foods, if there is a large formation of ice crystal, which is mainly because when we are slow freezing. So process when the uh, freezing is slow, so what will happen? Large disruptive ice crystals are formed, so which are mostly extracellular and resulting into the damage of the tissues. Whereas when we are doing fast freezing, the smaller or the fine ice crystals will going to form. So there will be fine crystals will going to form, and that will evenly distributed both sides and inside and the outside of the cell, and leads to the quality of the product to be better preserved due to less damage of the tissues. Therefore, the control will be there for the crystallization process, so that the freezing of the ice or the formation of the ice crystal can be properly achieved, and there will be less damage to the tissues. So fast freezing prompt prevention of the microorganisms also. In slow freezing, microbial growth is not prevented promptly, and the water vapor gradients will not be there. Hence, minimum dehydration will be there. Whereas ice crystals in the slow freezing have lower water vapor pressure, leading to dehydration of the product. So, slow freezing leads to large crystals, and large crystal causes the tissue damage. So, both are correct here. So, here in column 1, we have 4 methods of freezing: air blast, still air, sharp freezing, liquid immersion, and fluidized bed freezing. And in column 2, we have their appropriate statements like suitable for irregularly shaped food products, uses cold air at high velocity, submerges food in chilled liquid, and requires no fans or air circulation. So this question is very easy. Anyone who would like to answer this question? Anyone who would like to answer this question? So A is correct answer here. So basically air freezings are 3 types: still air shaft freezing, air blast freezing, and fluidized bed freezing. In this, basically packed or unpacked non-fluid foods, they will be frozen in the air at temperature ranging from -18 to -40 degrees Celsius. And in still air shaft freezing, here the products are placed in a very cold room where the temperature is -15 to -29, and the shaft freezers are basically cold storage rooms specially constructed to maintain low temperatures. And here no fan is required. Freezing time is 3 to 72 hours or more, depending on the condition and the size of the product. So here 2 is related to requires no fan for air circulation. Then air blast freezing, it produces the products, sorry, it places the products on the trays, either loose or in the packaging. Like this, all trays will be there, and the trays are placed on the freezing coils in the low temperature rooms with cold air blowing over the product. So there is fan required for air circulation. So here tray on this food product will be there. Below is the freezing coils, and above is the air. The temperature of the air is usually -28 to -46, and the air velocity is 15 meter per second. So it is related to use cold air at high velocity. Then is the fluidized bed. So in fluidized bed freezing, what happens? There will be the food product, like the solid food product, which can have different sizes, like peas, strawberries, and at the bottom, cold air will come, and this will be the mesh will be there. So air will pass, and based on the density, it will float. The food will float. So it can be form. This food will going to form the fluidized bed of the particles. Then because we have the forced air coming from the bottom, and it will lift the particles based on the density. So suitable for irregularly shaped food product. Finally is the liquid immersion freezing. So in this, what happens? Freezing of the food is there when we are submerging the food into very cold liquid and at the low freezing time. So sometimes we use nitrogen, liquid nitrogen for immersion. So liquid nitrogen is at very low temperature, minus 196 degrees Celsius. Sometimes we use liquid CO2, which is at around minus 78 degrees Celsius. And sometimes we use the brine solution. So in this, we use water and salt solution, and the temperature is around minus 10 to minus 20. And direct immersion, where we use the brine or glycol solution, in which we will food will going to directly placed into this liquids. So the liquid immersion freezing, it submerges the foods in the chilled liquids. So A is correct here. So uh, in the next week, we're going to discuss about food process, preservation and operation, food formulation and design, mathematical tools for food formulation, functional and designers of food, and 3D printed food for personalized nutrition. This we'll going to discuss in the next week. So the respective material will be uploaded, and if you have any doubt, please feel free to ask. This is the end of this today's session. If you have any doubt, please feel free to ask. So let's meet on next Sunday, same time. Thank you so much for joining the class. Anyone is having any doubt, please feel free to ask now.